have too many of one type and none of the other? Too many of any type? Yeah. No, so X and Y are both considered the sex chromosomes. The autosomes are 1 through 22, anything that's a non-sex chromosome, right? But there's all kinds of terms for, right, because the X has its own pair, right, it has a homologous chromosome because in a female, 2 X's. If you're X and Y, you're considered hemizygous. You're not really heterozygous. It doesn't really exist, but it has this other name. If you're boys, you're also heterogametic because you have 2 different sex chromosomes, whereas females have the same. Okay, so practice the practice problems for the 1-gene traits. I'll post the key later if I can remember to do that today. Or somebody email me if it doesn't pop into Moodle, in case you need to check your answers. If you want more practice, all you have to do is Google monohybrid genetics or Mendelian genetics quiz or Mendelian genetics problems, and you'll get a cajillion different sites where you can go either take quizzes or do problems with answers, find worksheets, all kinds of crap if you want to practice, you know, like the stuff that was on the quiz today. Right? If those all gave you a hard time and I'm not letting you see them, there's a billion questions out there that might look very familiar to you. Where do you think I get my questions from? For quizzes, right, that are out there. And if you don't get it, then you can print those, come into office hours, we can work through it or something like that. Okay, so there's lots of resources out there. I'm not trying to hide so you don't get a lot of practice. I just need to be able to sort of recycle the same questions because it's a lot of work just typing in questions and all that stuff for quizzes. Not to be mean. Yeah.
On the test, can you test questions like how we were doing it earlier, where there was there's plenty of who that was, and the only way you can get out of it was
yeah, there's likely to be one question about paternity and blood typing. No, I won't be that hard. I would do that to my 360 people. It just popped into my head and that just made me excited. But yeah, I won't do that to you guys. So it'll be like you'll know mom and baby and something about dad, you know, yeah. Is that cool? You guys are cool with those? And same with these 2-gene trait ones. You can search that online as well for extra practice. There should be a practice sheet posted for today for these times. Some are a lot harder than they need to be because I just pulled them from genetics textbooks. But if you can work through them and understand it, looking at your notes with help, then you'll be fine on the really hard ones. And they're all essentially based on the same monohybrid rules that we've already learned. Okay? It's just we're talking about 2 genes at the same time instead of doing them separately. If you do a 2-gene cross separately, you're going to get the wrong answers, because they're dependent on each other and who gets what, okay? So I'll go through these and then we'll do at least a couple examples, I think. And so when we're talking about, and how do you know if I'm talking about 2 traits or 1 trait? You're actually, physically, I'm telling you the phenotype about 2 different things, right? Tall and short is 1 phenotype. 2 alleles, 2, you know, types of the same gene, height. But if I'm talking about the plant is tall and green, or tall and yellow, or wrinkled and stinky, I don't know, right? That's 2 different things. So, you know, in the paragraph listing what we're talking about, if it's giving you 2 things, you ought to, 2 genes. I need to keep track of these. Some of them will be easy, and I'll tell you the genotypes. You'll get the phenotypes and the genotypes. You just have to do the cross. The other ones, you'll get phenotype. You have to figure out the genotype of at least one of them, or, you know, heterozygous at both traits. What does that mean? Big T, big R, little r, right? Heterozygous, homozygous for tall, heterozygous for color. Homozygous would be big T, heterozygous for color, big G, little g. Right? To be able to think about phenotypes, what is their genotype? I give you the genotype, you tell me the phenotype. Okay? And so it looks, right, something like that. And so color and shape are two totally different traits. So color, we're using one alphabet letter, capital for dominant, lowercase for recessive, just like we did for the one gene traits. The other gene, a different letter, big R for the dominant, little r for the recessive. So we know, because we're using two different letters, that it's two different genes. If it were just multiple alleles, right, we'd have, Y superscript A, Y superscript B, something like that, if it was just multiple alleles. The only multiple allele one you guys are going to get is blood type, so you don't have to worry about, that sort of thing. You'll recognize the blood type questions, those have multiple alleles. Everything else we do is just going to be two alleles. So two alleles for each, two genes. So if they're heterozygous for both traits, right, they're going to look dominant for both traits. Right? Because the heterozygote's under complete dominance, which all these are going to be under that umbrella again. As long as you have one capital letter of that gene, it looks dominant. And you'll either be told which one's dominant or which one's recessive, or you have to figure it out. How did we figure out which one was dominant and which one was recessive for a one-gene cross? Do you remember what that setup had to be? A test cross, exactly, to look at which one of one parent phenotype, the dominant phenotype, is heterozygous or homozygous. But if we're just given, remember, purple and white, yeah, we want to look at the F1. We set up the parentals, right? We make the two true breedings, and then whatever shows up phenotypically in the F1, the heterozygote, that's the dominant phenotype. So it's the same for a two-gene cross, except we're just looking at both, what am I looking at? Color and shape, right? So we'd set up the parental, which has to be true breeding. Right? And so one parent could be true breeding dominant, the other could be true breeding recessive. In this case, it really doesn't matter if one parent is dominant for one and recessive for the other. They don't have to both be dominant and both be recessive to be considered parental. Because if you cross these two or these two, you end up getting the same ratios. They'll be in different squares in your Punnett square, but when you count them up, it'll be the same. It'll end up looking the same. All of these guys are going to be heterozygous. They're all going to end up big R, little r, big Y, little y. Like, that's all they can be. That's all the parents have to give away. Okay. By convention, we usually show them like this, but not to confuse you that it can't look like that. Right? As long as they're opposite, the parents, this big R's, little r's, big Y's, little y's. You can't have, if there's big R's over here, you can't also have big R's over here, because then that's not the parental cross, because you want dominant recessive. Okay. So to make our Punnett squares, this is where sometimes people get a little crazy. Okay. It's the same idea as for the monohybrid, but we have to deal with two different genes and the combinations of gametes that could be made from that. So, okay, let's go back. Let's squeeze this up here a little bit. Whoa. Okay, so this guy down here is saying, okay, that if we cross either of these parentals, we get the heterozygote, right? So they're showing us here. All the offspring in the F1 are heterozygous. And because they already told us which ones are dominant and recessive, we know this guy is yellow and round, right? Because we told you that. But whatever that guy's phenotype happened to turn out is whatever's dominant. So if we weren't told, whatever that guy looks like, now we know which is the dominant trait. And then if we want to do this cross to get to that F2, we have to figure out, well, how do we set up our square? What are the gametes? Right? When we just had this guy, we knew half the gametes were going to be big R and half were going to be little r. And then that's, we just put those, and then the other one, right? And that's how we set up our square. Really, the things on the side are the gametes, things on the top are the gametes, and then the square helps us do all the different possibilities. Right? That's all the stupid squares for. When we have these two, we have to consider both of those genes and all the different combinations of gametes. And so this is saying, here, if we have this big Y and this big R could get together into a gamete. Right? If we're thinking about meiosis, they're on two different chromosomes. One goes this way, one goes that way, or the other one can go this way. Right? Or this Y could have been on the chromosome that gets stuck with that guy. So this one and this one could have gotten together in one of the gametes. This guy and this guy could have gotten together, this guy and this guy. If you need to set up meiosis using these two different homologous chromosome sets and do that, and have them labeled on the chromosomes, do that, so that you understand how all four of these gametes could form. Okay? Because that's just independent assortment, that's just meiosis. So all the combinations could happen in this one individual to make the gametes. This is just making gametes. We haven't started talking about making the offspring yet. Like, that's just what we line up on our Punnett square. And then the other parent obviously is the same genotype because it's F1 on F1. And so the other parent would make these exact things. And you'd end up with a square with 16 squares in it, which is a lot of squares. And so that's what they show us here, right? Here's all the gametes. Big R, big Y, big R, little y, little r, little y, little r, big Y. All the possibilities, those four, lined up on a Punnett square. Okay? And then filled in for you, because that would take me way too long. It'd take me all classes to fill that out. Okay? But again, it's over and down, right? One big R from this guy, one big R here, means that guy's big R, big Y, big Y, big Y, big Y. So this guy is homozygous dominant for both traits, right? He looks round and yellow. And you can follow all of these down and across. So you build the square just like you do with two gametes each, but there's four possible gametes. This, and again, just like the monohybrid, no matter what two heterozygotes with a two-gene cross you put in there, you will always get the same ratios in the square, right? Remember for the one gene square, it was 3 to 1. You got one homozygous dominant, two heterozygotes, and a homozygous recessive from big R, little r, big R, little r. A 3 to 1 phenotypic ratio, 1 to 2 to 1 genotypic ratio. It's the same thing here, except we don't worry about the genotypic ratio because there's too many of them, so we don't care, okay? We care about the phenotypic ratios. And the phenotypic ratios then tells us the genotypic ratios. So what the hell do I mean by that? If we go through here and we count all of the double dominants, so look at your guy and tell me how many in this big square are both round and yellow. You bet. So everybody count just so it makes sense to you. Right? So 9. So there's 9 that we know have at least one big R, right? To look dominant, you have to have at least one big R. To look round, yeah. And at least one big Y to be yellow. So the phenotype is round and yellow, and there's 9 of them. 9 out of how many is in this square? Yeah, 9 out of 16. And we'll just keep that in mind. So though all the possible combinations, if you look at all these, big R works, big Y works. Big R, big Y, little r, big R, little r, big Y, all the different possible combinations, as long as we have at least one big R and one big Y, we know they will look round and yellow. They have to. Okay? All right, now let's count up one trait that's dominant and one that's recessive. So let's go with green is the recessive and round is the dominant. Yeah. Okay, that's so good. And so what does that guy have to be? What did I said green, right? And round. So the round has to be exactly capital R. Yeah. So look, oh shoot, that's not the right letters. I made G's for green. Right? Does that make sense? Anything little y is going to look green because that's recessive. And what about the next category? What about the opposite? What about, yeah, wrinkled and yellow, you're old wrinkly. Right? That's the genotype. So that one's easy. It's either little r, big Y, or little r, big Y, little y. Right? There's only two options for these guys, but there's a whole crap load of different options for the 9s. And then what's the lonely little one? He's the big loser? Yeah, the double recessive. Little r, little y, little y. So the phenotypic ratio is always 9:3 to 3:1. We know what the 9 means, right? At least one big R, one big Y, at least one dominant of each trait. We know both 3 categories have to have one dominant, one recessive, the other dominant, the other recessive. And the 1 is always homozygous recessive. So that one's easy. The phenotype, there's only one of them, and there's only one genotype of that guy. Okay? So you should never, ever have to draw out this square, especially on a test, because it's going to take a long time to draw 16 squares and fill all that out, and then count up when I ask you how many are going to be dominant for one trait and recessive for the other. All you need to do is remember this and go, 3 out of 16 are going to be dominant for one and recessive for the other, and then another 3 is recessive for one, dominant for the other. Dominant, 9 out of 16. Recessive, 1 out of 16. Like that will never change. It's like big G, big T, little t. Doesn't matter. Big R, big Q, little q. Heterozygous on heterozygous. And F1 cross onto F1 to get to the F2 is always this. Always 9:3 to 3:1. Okay. You can draw it out at home just to make sure you trust yourself. Right? And you believe me that this is true, but I swear it's true. Okay. So I'm going to ask you questions about that, but they're going to be questions about 9:3 to 3:1 and which genotypes fit in which categories and that kind of stuff. Right? So that honestly is a memorizing thing. Right? Like I said, memorizing is not bad, it's just not all we should do. Right? But that's something we need to memorize and then understand why those numbers are what they are. Okay? But for most 2 gene crosses, especially ones I'm going to ask you something about how many offspring do whatever, as long as one of them is homozygous, your square is never going to be 16 squares because some of the lines will be redundant, right? And that goes back to when we talked about, remember when I was making fun of them because they only put one R in one and they just did that. And I was like, that's confusing because what happened to all the other gametes? Well, they really do exist, right? You can put both, but you're still going to get the same ratio. They're still 100% big R, little r. Okay? And so that's the same with the 2 gene. If there's anything redundant, you don't have to list it as a square because it's a ratio, so it's not going to change 50-50 or 75-25. So what do I mean by that? I mean, if you have this kind of cross, right, big T, little t, then this guy and that guy, which one goes where? This one. So, here, let me get rid of that because that's messy. So So this parent is up here. His possible offspring are big T, big P, or big T, little p, right? So isn't that all the gametes that guy could make? But he'll make two big T, big P's and two big T, little P's. Okay, but you don't have to list out that extra big, right? We don't need to put that one there. Or this one, because it's exactly the same as the one's already listed. We could, but we don't have to. And same with this one, right? Big T, little p, little t, little p, well, big T with the other little t with the other little p. Right, we could add that, and now guess what? We're back to our 16 squares. But the ratio is going to end up being the same if we just do these as if we do all of them. So we're not going to waste 5 more minutes on an exam filling in a bunch of squares that don't matter. Okay? So if you have a redundant gamete, we're already showing that one. We don't need to show this one. Half are big P, half are big T, little p. So we don't need to put those guys in there. Same with this one. Half are big T, little p, half are little p, little t. We don't have to show them because this still represents half and half. Half and half. So if you fill that out, right, this is what you get in the squares, and we would say, how many different genotypes are in this? 4, right? All are different genotypes. So what about phenotypes? We don't really know what they stand for, but as far as recessive and dominant. Yeah, 2, right? This is double dominant. This is double dominant. This is dominant recessive. Oops, I guess I could do a different color. And this is dominant recessive. So genotypes, we'd say 1 to 1 to 1 to 1. Phenotypes is just 1 to 1, right? Or 1/4, 1/4, 1/4, 1/4. 1/2 to 1/2. What's next? Okay, then I just wrote that out for you. See how nice I am?